Welcome to Ask Prep, your trusted companion for NEET and JEE preparation. Today we will study about a very important topic of biology, which is biotechnology, principles and processes, recombinant DNA, tools. Biotechnology is the use of living organisms or their components to make useful products or processes. It's not just about using microbes to make yogurt or cheese. Modern biotechnology involves using sophisticated techniques like recombinant DNA technology, where scientists deliberately modify the genetic material of organisms to give them new abilities. Think of it as genetic tailoring. We are cutting, pasting, and modifying genes to create organisms with traits we desire. Recombinant DNA technology, often called genetic engineering, revolves around combining DNA from two different sources. Imagine taking a gene responsible for insulin production from humans and inserting it into bacteria. These bacteria, once equipped with the human gene, start producing insulin, which we can then harvest for medical use. But how exactly do we do this? What are the principles and the tools involved? First, the central principle behind recombinant DNA technology is the universality of the genetic code. This means that the genetic instructions written in DNA are the same across almost all organisms, whether it's bacteria, plants, or humans. This allows a gene from one species to be read and expressed by another species if inserted properly. However, to achieve this, we need precise tools and carefully designed steps. Let's start by understanding the main tools. The first essential tool is restriction enzymes, also called molecular scissors. These enzymes can recognize specific sequences in the DNA and cut at those points. Each restriction enzyme has a unique recognition site, usually a short palindromic sequence, and it makes cuts that create sticky ends or blunt ends. Sticky ends are overhanging sequences that can easily pair with complementary sequences, making them very useful for joining DNA from different sources. Once we have the desired gene cut out, we need a carrier to transfer this gene into the host organism, and that's where vectors come in. Vectors are DNA molecules that can carry foreign DNA into a host cell and ensure it gets replicated. The most common vectors are plasmids, which are small, circular DNA molecules found in bacteria. Another important type of vector is the bacteriophage, a virus that infects bacteria. These vectors are engineered to carry the gene of interest, and they often contain special features like selectable markers, which help identify cells that have successfully taken up the recombinant DNA. Now, to join the foreign DNA with the vector DNA, we use another important tool, DNA ligase. This enzyme acts like glue, sealing the gaps between the foreign DNA and the vector DNA, creating a stable recombinant DNA molecule. Without DNA ligase, the pieces of DNA would remain separate and unstable. Once the recombinant DNA is ready, we introduce it into the host organism, typically a bacterial cell, in a process called transformation. But it's not as simple as just mixing the DNA with the bacteria. Bacterial cells need to be made competent, meaning they need to be treated so they can take up foreign DNA efficiently. Methods like heat shock or using calcium ions are commonly employed to make bacterial cells competent. After transformation, not every bacterial cell will have taken up the recombinant DNA. That's where selectable markers on the vector come in handy. For example, if the vector carries an antibiotic resistance gene, we can grow the bacteria on a medium containing that antibiotic. Only the bacteria that have successfully taken up the recombinant DNA will survive and grow, allowing us to isolate them easily. But the process doesn't stop there. Once we have bacteria carrying our gene of interest, we need to ensure that the gene is expressed correctly, that is, the protein product we want is actually made. This involves providing the right signals, like promoters and terminators, on the vector, ensuring that the bacterial machinery reads the inserted gene and produces the desired protein. Let's take an example to make this clearer. Human insulin was one of the first and most famous products made using recombinant DNA technology. Scientists isolated the gene for human insulin, inserted it into a plasmid, and then introduced this recombinant plasmid into bacteria. These bacteria, once transformed, began producing human insulin, which could then be harvested, purified, and used to treat diabetic patients. This method revolutionized medicine because it allowed the production of pure human insulin in large quantities, without relying on animal sources. So, as you continue your preparation, remember that biotechnology is not just a set of techniques or terms to memorize. It represents a profound shift in how humans interact with the living world. Understanding recombinant DNA and the tools involved opens a window into a future where science and innovation can solve some of the world's greatest challenges, from disease and hunger to environmental sustainability. Now, let's talk about the host organism. Most often, the host is a bacterium like E. coli, which is easy to grow and manipulate. In some cases, yeast or even plant and animal cells can be used, depending on the complexity of the gene expression needed. The choice of host depends on many factors, including the nature of the gene, the protein to be produced, and the final application. So, to sum up the journey of recombinant DNA, first, we isolate the gene of interest using restriction enzymes. Then, we insert it into a vector using ligase. This recombinant vector is introduced into a host cell, which is then selected and cloned. The final product is a genetically modified organism that can produce proteins or express traits not naturally found in its genome. Thank you so much for being a part of the Ask Prep community. We truly appreciate your time and dedication to learning, especially when it comes to mastering biology, a subject that's not only essential for exams like NEET and JEE. If you found this video helpful, please give it a thumbs up, share it with your friends who are also preparing, and make sure to subscribe to our channel so you never miss an update.